In this third example, we're dealing with 4 genes again, but now it's a little more involved because we do have a couple of them that are giving us 50% recombination frequencies. So the approach is going to be the same way as before. We're going to start out drawing a chromosome here, and I'm going to start with the genes that are located the closest together, which is going to be A and D here. So I'm going to put A and D right here in the middle. They're going to be 13 map units apart. Then I'm going to go to the 2 that are next closest, which is C and D. So I know that C and D are 24 apart from each other. So C could be 24 in either direction. If we look at the fact that A and C are supposed to be 37, that tells me that this must be the right location over here for C, because that's going to give me the 37 that I'm looking for between the A and C. So I'm going to get rid of this one. If I look at A and B are supposed to be 41. So that means that B could be 41 this direction, or B could be 41 this direction. And now I'm down to my 50%. If I consider that B and C are supposed to be 50% or 50 or more map units apart from each other, that does rule out this location for B. Because if B was located right here, it would be way too close to C. So instead, B is going to be located over here. That will give me the 41 from A, and that will give me the greater than 50 from C. We can actually come up with the actual distance if we add together the 41 plus the 13 plus the 24 there. That would give us the actual map units between them. If we look at B and D, we can simply just double check here, make sure that between B and D, when we look at it, we do end up with greater than 50, and we do. So the correct answer for this one would be B, A, D, C, or C, D, A, B, however you want to write it. Okay, so make sure that you can create linkage maps, order genes on a chromosome, and do go through some of those practice questions that you do have on Angel. Now, if we talk about some other things that can result when we're talking about individuals and chromosome numbers. It is possible to have abnormal chromosome numbers. We said previously that each species has kind of a set characteristic number of chromosomes, but that does not mean that every single organism of that species has that exact number. So for instance, 46 chromosomes is what we consider humans to have. But there are some humans that have 45. There's some that have 47, 48. So it does happen. If we look at how these abnormal chromosome numbers come about, it really does come from errors during meiosis. These errors during meiosis are called nondisjunction. And what that nondisjunction means is it is a failure to separate. Now this failure to separate can happen either in meiosis 1 or meiosis 2, and it could be a failure of the tetrad to pull apart, as we see here in meiosis 1. If that is the case, you end up with too many chromosomes on one side compared to what we have over here in this dividing cell. So in the end, you end up with some gametes that have too many chromosomes and some gametes that have too few. If we have the nondisjunction happen later on, so in meiosis 2, we don't get the pulling apart of the sister chromatids, and in this case we get a few gametes over here that are normal, and we get one that has too many, one that has too few. So keep in mind that our gametes are supposed to be haploid. Well, the reason we have 46 chromosomes in humans is because we have an egg that has 23 and a sperm that has 23, and we put the 23 and the 23 together, we get the 46. Well, what happens if we now have an egg that has 24 and we have a sperm that has 23? We're not at 46 chromosomes anymore, so now we're up to 47. So keep in mind that when we have fertilization, it is random fertilization, which means a random selection of an egg and a random selection of a sperm that are going to unite with each other. If a gamete that has had nondisjunction occur is chosen for fertilization, then the individual that we get is going to be a little bit off in their chromosome number. So these individuals are what we call aneuploids. Aneuploidy results from fertilization using gametes in which nondisjunction has occurred. So this is a condition in which the offspring would have an abnormal number of a particular chromosome. So this would be humans that do not have a total chromosome number of 46 anymore. And keep in mind, we're supposed to have 2 of every chromosome. So they might have 3 of a particular chromosome or 1 of a particular chromosome. We can classify aneuploid conditions based on those off numbers. Trisomic conditions are where we have 3 copies of a particular chromosome. For a human that is trisomic, they would actually have a total chromosome number of 47. We can also have the monosomic condition where there's only 1 copy of a chromosome, of a particular one, and that would be a human that has 45 chromosomes in it. So if we look at a number of examples of aneuploid conditions in humans, they are associated with a number of serious human disorders, and the most common one is Down syndrome. Down syndrome is the result of having an extra chromosome 21. So if you look at this karyotype here, you can see that there are 3 chromosome 21s there in this individual. So for someone with Down syndrome, they have 47 total chromosomes. This is the most common or most obvious trisomic condition or aneuploid condition in humans because these have a definite phenotype to them. They look different, and they also have a fairly lengthy lifespan. So we do fairly commonly see individuals walking around that do have Down syndrome. Now, that's not to say that this is the only trisomic condition or aneuploid condition out there. Trisomy 13 is an extra copy of chromosome number 13. With this one, there is severe mental retardation and developmental delays, and only 5% of these individuals live past their first birthday. Okay, so the reason we aren't as familiar with trisomy 13 is because there are not very many surviving individuals with this condition. Trisomy 18 is another similar one where only 10% of these live past their first birthday. So again, you're not seeing a lot of surviving individuals here, which means we're not as familiar with this one. Notice we have 3 examples that we've gone through of trisomic conditions, and all of them have pretty severe phenotypes. Even with the trisomy 21, which is our Down syndrome, even though there's a fairly lengthy lifespan there are significant phenotypes associated with that. Now, we don't have many examples of trisomic conditions in humans where we're talking about the autosomes. So notice that all 3 of these that we went through were examples where the number of autosomes was incorrect. They result in very severe phenotypes. So severe that if there was perhaps a trisomy 1 or trisomy number 3, those are not individuals that are surviving. So those may be miscarried babies. So we're not getting production of the individuals with those conditions. That's not to say it can't happen in the actual fertilization and come up with the zygote, but it's not going to develop fully. Now, if we switch pace a little bit and we talk about nondisjunction with the sex chromosomes, this can certainly happen as well. So when nondisjunction occurs, it is a random selection really of which chromosome it's going to happen with. So if it happens with an X or a Y chromosome, we can also get aneuploids. Now this is going to be a lot more common. So Klinefelter's syndrome is the most common example we have of aneuploids with off numbers of the sex chromosomes. This is when you have an extra X chromosome in a male, so they are XXY. So they have a total number of chromosomes of 47. And if you look at the frequency here, this is 1 in every 500 males, and most do not have any symptoms whatsoever. So most of them do not even know that they have Klinefelter syndrome. There are sometimes some hormone abnormalities, slight hormone abnormalities, so that can result in some weak muscle development, perhaps someone that's a little taller and more lanky. But that is not necessarily a real good phenotype here because we have lots of individuals that are tall and lanky, and that doesn't mean they have Klinefelter syndrome. With these, because the hormones sometimes are a little bit off, there's often a fertility issue. So if someone later in life was having some fertility issues, if they went to a specialist to have that investigated, that might be when they find out that they actually have Klinefelter syndrome. So notice the difference here in someone that has an extra copy of an autosome and they either do not survive or they have severe phenotypes versus someone with an extra copy of a sex chromosome and hardly can tell the difference. If we look at an example in females, Turner syndrome is when a female is missing all or part of one of her X chromosomes. So this is our monosomic condition. With this individual, she would have 45 chromosomes. And the severity of this one varies a bit depending on how much of that second X they're actually missing. So in this karyotype here, we see that the person is missing the entire second X. That would be a severe copy of, or a severe situation of Turner syndrome. If we're only missing a tip off of the second X, then that's not going to be as bad. This is 1 out of every 2,500 female births. With this one, you do have a little bit more phenotypes associated with it. And these usually, because of the poor ovarian development, they are going to be infertile. Here we just have a list of a few more examples of aneuploid conditions with off numbers of the sex chromosomes. The take home here is to notice that they really are present in fairly high frequency. Keep in mind that these individuals do not have the severe phenotypes that we had with the abnormal numbers of the autosomes. Okay, so nature really has established a very careful balance of the autosomes, and 2 seems to be really the right number. Anything less than 2 or more than 2 is not going to be a good thing for the individual, and in most cases is not going to be an organism that survives at all. Polyploidy is when we're looking at more than 2 complete sets of chromosomes. So basically, before we talked about diploid, here we're talking about more than diploid. So we can have triploids, which would be 3N. That would be 3 copies of every chromosome. We can have tetraploids, which is 4N. That one right there is probably the most common of all of the types of polyploids. We can have hexaploids, which would be 6N, octoploids, 8N, and so on. So hopefully you're getting the idea. We can have quite high numbers of chromosomes or chromosome sets. Now this is very unusual in animals. There are a few examples of this in animals, but it is very unusual. In plants though, this is very, very common. So many plants are polyploids. Many of the commercial varieties of plants that we consume are polyploids. For example, bread wheat is a hexaploid. Barley is a polyploid. A lot of those cereal crops that we eat are going to be polyploids. So make sure that we do see the difference between an aneuploid, where there is one particular chromosome where we don't have the right number of them, versus polyploids, where we're talking about extra copies of every single one of the chromosomes. There can also be alterations in chromosome structure that occur within organisms. So these alterations in chromosome structure can happen at numerous times during a cell's life cycle. These could happen during the DNA copying process. They could also happen during the crossing over process. If you look at the first one, it's a deletion. So in this case, we're missing an area on the chromosome. We would assume that that area has some pretty vital information on it. So that probably is not going to be a very good thing to be missing that section, which we're calling D in this slide. The next one, the duplication, results in extra information on the chromosome. Now, many times the cell will be able to just kind of turn off that extra area, similar to what we did with the X inactivation, and maybe that's not going to present a problem at all. In any case, we're not missing any information. The inversion, we're just getting an area turned around in the other direction, so everything is still present. And then the last one, the reciprocal translocation, we're swapping ends of the chromosome. Now we want to keep in mind that that would not be considered crossing over here, because this is occurring between non-homologous chromosomes. We're not swapping the same type of information. Again, though, we still have all of the information present, and it's still present in the same number of copies as it was before. Now, with these bottom two, they're not normally a problem for the individual that has them because they still have the right number of copies of everything. Where they can become a bit of a problem is during the gamete production in that individual. So keep in mind that during meiosis, the chromosomes, the homologous chromosomes, need to pair up with each other. So we need that synapsis process to occur. Well, if you have one chromosome that looks like this one, and you have one chromosome that's like this one, they have a hard time pairing up exactly with each other because no longer do they contain the same contents. So that affects the crossing over process, and we get a lot of abnormal gametes resulting. So many times there are fertility issues in individuals that have those particular chromosome rearrangements. Another thing we want to talk about is called genomic imprinting. Now, genomic imprinting falls under the field of what we would call epigenetics a lot of times. Genomic imprinting is when the phenotypic effects of certain genes depend on which allele is inherited from the mother and which allele is inherited from the father. Now, previously, we didn't really worry about which allele we got from mom or dad. We just looked and said, okay, we've got a big A, little a, or two big A's, two big B's, and that was that. Well, if you look here in this slide, notice that if we cross these two mice with each other, we're crossing a green one with a blue one. In both cases, but on the top one, all the mice offspring are green, and in the bottom, all of the mice offspring are blue, even though in both cases we're crossing a green with a blue. Well, the reason is that we've got this genomic imprinting going on. So notice that they are looking like dad. So on the top one, dad was green, the offspring are green. Bottom one, dad was blue, the offspring are blue. So this involves what we call the silencing of certain genes that are stamped or imprinted during gamete production. So in other words, in the mom, in this case, this gene for mouse color is getting stamped or imprinted. And when a gene is stamped or imprinted, that basically means that it is turned off. Or in other words, we can't use it. So if mom is basically turning off the gene in her eggs, when it passes on to these offspring, they're not going to be able to use it. So they will be using the copy they got from dad as kind of a default here. If we look at an example here, this is a mouse size gene. So we have a normal version and we have a mutant version which produces a dwarf mouse. So the top mouse has two copies of the normal version, and we expect it to look normal, which it does. The other two examples are both heterozygous mice, so they really do have the same genotype, but notice that they do not have the same phenotype. So one is a normal sized mouse and the other one is a dwarf mouse. The reason for this is because the mother's copy is imprinted or stamped. So that means that the mother's copy is kind of turned off. So it's there, but the offspring can't really use it. So that means that they work off of the father's copy, whatever it happens to be. So for this mouse, it gets to work off the normal copy. For this mouse down here, it's working off of the mutant copy, and now it is dwarfed. This involves chemical modifications on top of the DNA. So the DNA sequence is the same, but we're adding extra chemical groups on top of it that prevent the cell from really utilizing that particular section of the DNA on, say, the mother's chromosome. In this example, it is the mother's chromosome that's being imprinted. It is not always the mother's that's imprinted. Sometimes it is the father's. It's very gene specific as far as which one is getting imprinted. This field of epigenetics is a very kind of hot area of research because, as you can imagine, the information is there. So if we could figure out a way of kind of reprogramming them to use the version of the gene that we want them to, there might be some benefits to that. There are some human genetic disorders that genomic imprinting plays a role. So if you can imagine that someone was heterozygous, they have one good copy and one bad copy, we would want to make sure that they're working off of the good copy. Okay, many times they're not. So that might potentially be one way to cure some particular diseases. The last thing we want to talk about in this chapter is the inheritance of organelle genes. All of the inheritance we've talked about so far has been nuclear genes, or genes that are located in the nucleus. So that was all based on meiosis. Well, keep in mind that we do have some genes that are not in the nucleus. So these would be in the mitochondria and the chloroplast. And in this case, it is going to depend solely on the maternal parent. So this means that we inherit all of our organelle genes from our mother. The reason for that is really right here as you see in this picture. The large structure would be the egg, so that's a human egg, and we've got all of these human sperm that are around the outside that are trying to enter in and fertilize that egg. So the egg is from the mother, obviously. It has a nucleus in it. It contains 23 chromosomes from the mother. But in addition to having the nucleus, it also has a bunch of cytoplasm, which came from the mother, which contains organelles, such as the mitochondria or the chloroplast if we're talking about plants. So when the sperm comes in there, the sperm is really just bringing chromosomes to combine with the chromosomes from the egg. But once the sperm enters and brings the chromosomes, now we call this structure a zygote. And that zygote contains cytoplasm from the mother, which contains organelles from the mother. So 100% of our organelle genes come from our mother. We do not inherit any from our father. This makes organelle genes very useful when we're talking about studying pedigrees or studying whether or not people are related to each other. If you're talking about organelle genes, they're not changing every generation in the same manner that the nuclear genes are. So the nuclear genes, we're kind of combining half and half from two parents, which switches things up a bit. And in addition to that, we're recombining the genes from our mother and from our father when we go through the crossing over process. The organelle genes are passed on in just an exact copy. So our genes, as far as what we get in our mitochondria, would be the exact same as all of our siblings. They would be the same as our mother. They would be the same as her siblings, as her grandmother's siblings. So hopefully you're getting the idea here that they are a good way to kind of track families. Another thing to keep in mind, though, is that there are some genetic disorders that are carried on DNA in the mitochondria. So in that case, a mother, if she had one of those disorders, she would pass it on to every single one of her children, no matter what. So 100% of her offspring would get it. However, if a father had a disorder located on a mitochondrial gene, he would not pass it on to any of his offspring. So just make sure we understand that if we're talking about organelle genes, the inheritance comes strictly from the mother.